Hello everyone and welcome back to my channel. In today's video, we're diving into the fascinating world of chimera. If you're curious about what a chimera is, how it relates to DNA replication, and the techniques involved in studying this intriguing subject, you've come to the right place. First, let's break down what a chimera is. In biological terms, a chimera refers to an organism that contains cells from two or more different zygotes. This can occur naturally, such as in the case of some animals and in human twins, or it can be created artificially in the lab for research purposes. Chimera studies can provide valuable insights into genetics, development, and various biological processes. Now, I'll walk you through the process of creating and studying chimeras, including useful techniques that are commonly used in the field. 1. Cell fusion techniques. One popular method to create a chimera is through cell fusion, where cells from different sources are combined to form a single cell with genetic material from both. Techniques like electrofusion and polyethylene glycol (PEG) fusion are commonly used. 2. Genetic engineering. To better understand the role of specific genes, scientists can use techniques like CRISPR-Cas9 to modify the DNA in one of the donor sources before fusion. This allows researchers to study the effects of gene expression in mixed cellular environments. 3. In vivo studies. For a deeper understanding of how chimeras develop, researchers often conduct in vivo studies, which involve creating chimeric organisms in model systems, such as mice. This helps us learn about tissue integration and the impact of chimeric cells on overall organism development. 4. Imaging techniques. Advanced imaging techniques, such as fluorescence and confocal microscopy, are critical for observing the structure and function of chimeric tissues. These techniques allow scientists to track the origins of cells in a mixed tissue environment. 5. Ethical considerations. As we venture into the realm of chimeras, it's crucial to address the ethical implications of this research. Discussions around chimeric organisms often revolve around biodiversity, animal welfare, and the potential for human chimeras. In conclusion, understanding chimera can provide significant insights into genetics and developmental biology. Whether you are a student, a researcher, or simply someone interested in science, I hope this video equips you with valuable knowledge and techniques to explore the fascinating world of chimeras further. The combining of goat DNA with human DNA would result in a hybrid creature known as a chimera. This organism would possess characteristics and traits of both a goat and a human. However, such genetic manipulation is currently illegal and highly unethical. In mythology, a chimera is a creature composed of parts from multiple animals, typically depicted as having the body of a lion, the head of a goat protruding from its back, and a snake for a tail. The term chimera has since been adopted in biology to describe an organism that contains cells from different species, resulting from genetic manipulation or fusion. In genetic research, chimeras can be created by combining cells or tissues from two different organisms, leading to an organism with a mix of genetic material. This process can be done by merging embryos from different species or by introducing foreign genetic material into an organism's cells. Chimeras have been used in scientific research to study embryonic development, disease mechanisms, and potential therapies. However, creating chimeras involving human cells is a controversial and ethically complex issue, raising concerns about the creation of creatures with human-like traits or capabilities. In summary, a chimera is an organism composed of cells from two or more different species, either naturally occurring or created through genetic manipulation. The term is derived from mythology but has taken on a scientific connotation in the field of genetics and biology. Creating chimeras involves combining cells or tissues from different species, resulting in an organism with a mix of genetic material. This process can lead to a number of scientific changes, depending on the specific cells involved and the purpose of the research. Some potential scientific changes that can happen during the creation of chimeras include: 1. Study of developmental processes. Chimeras can be used to study developmental processes and tissue formation. By combining cells from different species at early developmental stages, researchers can observe how cells interact and differentiate to form different tissues and organs. 2. Disease modeling. Chimeras can be created to model human diseases in animals, allowing researchers to study disease mechanisms and test potential treatments. For example, human cells can be introduced into animal models to study how diseases affect specific tissues or organs. 3. Regenerative medicine. Chimeras have the potential to be used in regenerative medicine to generate tissues or organs for transplantation. By combining human cells with animal embryos, researchers can create animals with human-like tissues that could be used for organ transplantation in the future. 4. Drug testing and screening. Chimeras can be used to test the effectiveness of drugs and study drug metabolism in living organisms. By introducing human cells into animal models, researchers can evaluate how drugs interact with human tissues and organs. 5. Ethical and regulatory considerations. Creating chimeras involving human cells raises ethical and regulatory concerns, such as the potential for the development of creatures with human-like traits or capabilities. Scientists and policymakers must carefully consider the ethical implications of creating chimeras and establish guidelines for responsible research practices. These boards assess the ethical implications of the proposed research and ensure that it complies with ethical standards and guidelines. 2. Research guidelines. Many scientific organizations and institutions have developed guidelines for the creation and use of chimeras in research. These guidelines outline best practices, ethical considerations, and regulatory requirements for conducting research involving chimeras. 3. Legal restrictions. Some countries have laws in place that restrict or prohibit certain types of research involving chimeras. For example, there may be regulations that prohibit the creation of chimeras with certain genetic modifications or that involve the introduction of human cells into non-human animals. 4. Oversight and monitoring. Research involving chimeras may be subject to oversight and monitoring by regulatory agencies or government authorities. These agencies ensure that research is conducted responsibly and in accordance with established guidelines and regulations. Overall, regulations and guidelines play a critical role in ensuring that research involving chimeras is conducted ethically and responsibly. Researchers are required to adhere to these regulations to protect the welfare of research subjects and to maintain public trust in scientific research. Can chimeras be used to develop new treatments for diseases? Chimeras have the potential to be used in developing new treatments for diseases in several ways. Some of the ways in which chimeras can contribute to medical research and the development of therapeutics include: 1. Disease modeling. Chimeras can be used to create animal models that mimic human diseases by introducing human cells or tissues into animal hosts. This approach allows researchers to study disease mechanisms, test potential treatments, and evaluate the effectiveness of new therapies in a more accurate and relevant context. 2. Drug testing and development. Chimeras can be used to test the safety and effectiveness of new drugs or therapies. By introducing human cells or tissues into animal models, researchers can evaluate how drugs interact with human tissues and organs, predict potential side effects, and assess the efficacy of experimental treatments. 3. Organ transplantation. Chimeras hold promise for generating human organs for transplantation by combining human cells with animal embryos or hosts. By creating animals with human-like tissues, researchers aim to develop a sustainable source of organs for patients in need of transplants, potentially overcoming organ shortages and compatibility issues. 4. Regenerative medicine. Chimeras can be utilized to study tissue regeneration and repair processes by introducing human cells into animal models. This research may lead to advancements in regenerative medicine, such as developing treatments for injuries or diseases that involve tissue damage or loss. While chimeras offer exciting possibilities for disease modeling, drug development, organ transplantation, and regenerative medicine, there are ethical and regulatory considerations that must be carefully addressed. Researchers must adhere to ethical guidelines and regulatory frameworks to ensure that their work is conducted responsibly and respects the welfare of all research subjects involved. If you found this video helpful, please give it a thumbs up, subscribe for more content, and share your thoughts in the comments below. Did you learn something new today? What topics would you like to see covered next? Thank you for watching, and I'll see you in the next video.